Okay, we're going into chapter 9 tonight, and I'm sure many of you have lost lots of sleep wondering things like why cells divide, when cells divide, how they divide. We've studied all kinds of different types of cells over the last few weeks. Is cell division the same for all cells? For instance, prokaryotic, eukaryotic, etc. You've probably wondered how often they divide. How do they get all the right stuff in both cells? And some of you may have even wondered what happens when cell division goes wrong. How many people lose sleep over these questions? Okay, so we're talking about cell division tonight. We'll start with unicellular organisms, those organisms that are one cell. An individual is one cell. We're not going to spend a lot of time on that because we're going to talk about that later in chapter 20 and 21. But when a single-celled organism divides to make two cells, that's actually how they reproduce. So cell division in single-celled organisms is asexual reproduction. We can really say that division actually multiplies the population. So division for these multiplies the population. So when a single cell, for instance an amoeba or a paramecium or some single-celled organism, divides to produce two, it's asexual, so those two that result would be genetically identical to the original cell. So if you continue to do that without mutation, all of those cells would be genetically identical. Just a little terminology before we go on. Some of you may have heard this terminology before, but when we talk about cells dividing, we oftentimes call the cell going into division the mother cell. And the two cells that result from that division, the daughter cells. Have you ever heard that before? The mother cell has nothing to do with gender. It has nothing to do with that cell being the mother in the terms that we think of it. It's just a terminology. The cell going into division is the mother cell, and those coming out are the daughter cells. Okay? Just didn't want you to get confused if you read that in the book or heard that. Okay? So let's go to multicellular organisms, as are you. Okay? So your cells, your body cells, divide for one of two reasons. Okay? When one of your body cells divides, it's either for growth, that means that you're growing. Okay? All growth is cell division. That's how you grow. Okay? Or if you're already grown, then your cells are constantly dividing to replace cells that die or are damaged or injured. Okay? So it's either growth or repair. So your cells, your body cells, divide for growth or repair. Your body cells we sometimes call somatic cells. You might want to write that in the margin of your notes, S-O-M-A-T-I-C, somatic cells. Somatic. Soma means body. Okay, so we sometimes call your body cells somatic cells. So what does this cell division function to do? Well, it keeps that surface area to volume ratio in check. It maintains that surface area to volume. You may remember going over this in chapter 4 when we said cells start out small, and I started on this side of the room. They start out small, and they grow, and they grow, and they grow, and then they get big. And we said that cell has to do one of two things. What? It either has to divide to produce two smaller cells, or it would eventually die because the plasma membrane could not maintain the contents of the cell. Remember, cells have to have a large surface area to volume ratio, and as cells grow larger, that surface area to volume ratio gets small. Okay? It also transfers a complete set of chromosomes to the daughter cells. Okay? So cell division functions to transfer a complete set of chromosomes to the daughter cells. I think that's so neat to think about, y'all. Think about it for a moment. At one point in time, many years ago, you were but yet an egg that was yet to be fertilized by a sperm. Okay? I know you don't like to think about it. Okay? But your mom and dad did it at least one time because you are here. Okay? When that sperm fertilized that egg and that genetic combination was present in that cell that we're now going to call a zygote, that's the first time that genetic combination has ever existed in the history of this planet. You are that unique. Okay? So those chromosomes that were present in that zygote were copied into 2 cells. And then those 2 cells divided and copied that same genetic information into 4 cells, and 8 cells, and 16 cells, and 32. And now we have you. So that same genetic information, with a few mutations because of age, that same genetic information is now in every cell of your body, except red blood cells. They don't have nucleus. Okay? So, a little bit of review just to make sure that we're all on the same page. Do you guys remember back in chapter 4 when we said that your genetic information existed as something that we called chromatin in a non-dividing cell? Do you remember that? And we said it was really diffuse and we can't see it. It was, when we looked at the illustration of the nucleus, it was those shadowy areas in the nucleus, okay? And I said it's very diffuse and it's stringy, but if we look deep down inside the chromatin, we see that, which we call the double helix, your DNA, okay? Then we said before cell division, your chromatin actually replicates itself and condenses, right? And that's what we called a chromosome. A little bit of review from chapter 4 there, okay? So each species has a particular number of chromosomes, okay? Does anybody know how many we have? 23 pairs. 23 pairs, 46 chromosomes, yes. Well, you know, that's a lot of chromosomes. I used to think when I first started studying science, like in junior high school, we got 46 chromosomes, and we're like the most advanced, as far as evolution is concerned, we're the most advanced, right? So we probably have the most chromosomes. Well, take a look at this. Guys, chickens have 78. We have 46. Take a look at that, okay? Tobacco has 48. Look at that. Wow. I mean, yeast. Think about this. Yeast has 32. They're right behind us, okay? So, I mean, obviously, it's not how many chromosomes you have. It's the information on those chromosomes that's important, right? But what is one thing that you notice about all of those numbers of chromosomes for those particular organisms? They're all even. That's exactly right. That's because chromosomes come in pairs, okay? We're going to call them homologous pairs, and they look alike. Those pairs actually look alike. They come in pairs. You said we had 23 pairs, right? So let's take a look at those 23 pairs. These are actual human chromosomes. Someone has taken a picture of a cell in division, or close to division, because remember, that's the only time we see our genetic information as a chromosome, right? After it condenses. So some scientist has stained and taken a picture of these chromosomes, and then he cut them out, and he placed the ones that look alike next to each other, and we call this a karyotype, okay? It's a picture of someone's chromosomes. This is one of the first things that a geneticist would do if you go for genetic counseling, or if you have some genetic issue in your family. The first thing they're going to do is to karyotype your family members. Okay? Just take a picture of your chromosomes. When they group the chromosomes into homologous pairs, those that look alike, what they're looking for are chromosomes that are the same size, When I say they look alike, they're the same size. And the centromere, or that structure which is holding the copies together, is in the same place for those two chromosomes that we're going to call homologous. For instance, in pair number 1, they're the same size, and there's, can y'all see that? My laser's dying. Can you see the laser? There's the centromere right there, okay? And for the different pairs of chromosomes, the centromere is going to be in a different location. Look at pair number 10. The centromere is right there. Look at pair number 16. The centromere is right there. They're really small. And poor old pair number 14, the centromere is at the top. Look at that. So the arms are different lengths, and that, depending on where the centromere is. So what they would do is they would group them together by homologs. The homologous pair goes together, and then they place them by size. So the largest one is always going to be pair number 1. The smallest is going to be pair number 22. And in a karyotype, the 23rd pair position is always reserved for a special pair of chromosomes called the sex chromosomes. So obviously, this example is a female because there's 2 X chromosomes right there. Had this been a male that had been karyotyped, there would only be one of those X's and there would be a Y. And I'll show you a Y later. It's very different from the X. Okay? So that's someone's 23 pairs of chromosomes. Okay, so if we said earlier in the semester, we're going to think about our DNA as a cookbook. Remember us talking about that? And so now we know that every cell has the whole cookbook. Okay? So if we think about these pairs of chromosomes, these homologs, we can almost think about the pair as a certain chapter in the cookbook. Okay? So the recipes that are on pair number 1 are not going to be on any other pair. Okay? For instance, if we have a gene or a recipe for eye color on pair number 1, then that gene is not going to be anywhere else. Okay? Each pair of chromosomes has its own unique set of genes. Okay, so let's talk about that gene for eye color on chromosome number 1. If these were your chromosomes, where did they come from? Your parents, right? So that means that half of these came from mom's egg and half of them came from dad's sperm. So it's going to be one from each pair. So one of the number 1s came from dad, one of the number 1s came from mom. Pair 2, one of them came from dad, one came from mom. Pair 3, one came from dad, one came from mom. So if your gene for eye color is on chromosome pair number 1, then you got a form of that gene from dad and a form of that gene from mom. And they're going to work together to make you who you are. Okay? Is everybody good with that? Okay, so when we have 2 that look alike, that condition, when we have the homologous pairs together, that condition is referred to as being diploid. Okay? When we have 2 of each kind, it's diploid. Okay? Di means 2. So, your somatic cells, what's a somatic cell? A body cell. Your body cells, your somatic cells are diploid. Okay? Learning lots of new vocabulary tonight. Okay, so what about that egg and that sperm we just talked about? Okay? We said that the egg would have had half, one from each pair, right? And the sperm would have had half, one from each pair. Well, when we only have one of a particular kind of chromosome, it's for that cell, that egg and that sperm, are referred to as haploid. So one of each kind is haploid. Okay, everybody good? Yes. Okay. Okay, so earlier in the semester we talked about how, when we were talking about cell size, we talked about the cell starting out small and growing and growing and growing and then dividing, and I even walked from one side of the room to the other. That period of time from when a cell comes out of division until the cell divides again is called the cell cycle. Okay, so tonight we're talking about the cell cycle. All of that time, all of the events that occur from when the cell is small and just came out of division until when it divides again is divided into two separate events. We have interphase and the mitotic stage. What does inter mean? In between? In between, yes, very good. So interphase is going to be that period of time in between divisions. It's going to be from when it came out of division until it gets ready to divide again, okay? And then, of course, the mitotic stage is going to be the actual dividing, okay? Interphase is divided into three parts, G1, S, and G2. And we're going to talk about what happens in all of these stages. The mitotic stage, which is sometimes referred to as the M stage, is divided into two events, mitosis and cytokinesis. Okay? And you have an extra blank. I've got to fix that. I'm sorry, I got blank happy. Okay. You have an extra blank there. Just make it work. Yeah, just mark that one out. Yeah. Well, you need to put one of them. Oh. Yeah. No, that was supposed to be the M stage, and that was supposed to be mitosis and cytokinesis. Yeah, you got it. Okay, everybody good? Okay, so let's look at this for a minute and talk about it. If a cell, when it's small and just came out of division, it goes into G1, and then it goes into S, and then it goes into G2, and then it divides into two cells. Those two cells that come out of division go into what? Back to G1. S, G2, they divide. Then what? Back to G1. S, right, exactly. So it's a cycle. It's called the cell cycle. So a cycle is like a circle, right? So let's look at it like a circle, okay, because that's really what it is. So in looking at this illustration, the blue part of this cycle is interphase. The green part is going to be the mitotic stage, okay? So let's look at that. The first part of interphase is G1. Okay, so where did the cells come from? When they went into G1, where did they come from? They just came out of division, right? They just came out of division. They went into G1, and then they're going to go into S, and then they're going to go into G, and then they're going to divide. Yes, M. And then those two cells that result from that division would go straight into G1, right? Is everybody with me? So the cell that just came out of G1, that just went into G1, just came out of division, right? So big cell divided into two little cells, right? So the first order of business in G1 is to grow. So the first thing those cells have to do that result from a division is get back up to size. G1 is all about growing. And think about it. You took one big cell and divided it, so each of those resulting cells, each of the daughter cells, got half of the organelles. So we got to make organelles. We got to grow. We got to get back up to size. Okay? So even before the cell goes into division, it's preparing for division. So S is replication. This is when your chromatin replicates. This is when your DNA makes a copy of itself. Then the cell will get ready to divide. And keep in mind, in division, something is going to have to tear that chromosome apart, right? I mean, that replicated piece of chromatin that now has condensed has to be torn apart and each half put into separate nuclei. The thing that's going to tear that apart and move it around for us are proteins. So G2 is all about making some proteins, getting ready for division, and continuing to grow. Then we're going to go into the M stage, which is the mitotic stage. What did we say it consisted of? Mitotic stage consists of mitosis and cytokinesis. Exactly. Mitosis is when the nucleus divides. You would have a nucleus that divides and produces two nuclei, each nuclei having its half of each chromosome. Cytokinesis is division of the cytoplasm. Cytokinesis is when you have one big cell, the plasma membrane is going to start pinching inward and pull those two cells apart, and you physically end up with two cells. So mitosis is nuclear division resulting in two nuclei. Cytokinesis is division of the cytoplasm resulting in two cells. What would happen if we had mitosis without cytokinesis? Anybody? No. You would have two nuclei. You would have one cell. You didn't have cytokinesis, so you still got one cell, but you have two nuclei. So you would have what's called a multinucleated cell, and some of your cells actually do that. They experience mitosis without cytokinesis, so you have one cell that has two nuclei in it, okay? Okay, so this whole process is under very, very strict control by lots of signal proteins called cyclins, okay? A signal protein is something that's going to tell the cell it's okay. It's a signal. Tell the cell it's okay to continue. These cyclins, and there's lots of them, these cyclins, the amount of these proteins, these signals, actually increase and decrease as the cell cycle continues, and that is a signal. The amount of cyclin in the cell is a signal to the cell telling it that it's okay to continue, okay? So cell division is under, cell division going from G1 to S to G2 to M is under very, very strict control. So the amount present increases and decreases as the cell cycle continues. There's a couple of really important checkpoints where the cell actually pauses to check. The cyclin will tell the cell to pause, and it'll check to make sure everything's okay. That's going from G2 to M and from G1 to S. Very important checkpoints for the cell. From G2 to M and G1 to S. So let's take a look at that, because these are the points at which, if something is wrong, the cell cycle will pause and allow the cell to try to repair the damage. And then we'll talk about what happens if the cell can't fix the problem, okay? So here's my cell cycle again, G1, S, G2, and M. I've got a checkpoint right there from G1 to S. Here's my checkpoint right there. I've got a checkpoint right here from G2 to M, and I've got checkpoints within mitosis, or the mitotic stage itself. So let's talk about why these are important, okay? Why is the checkpoint between G1 and S important? Well, think about it for a moment. Where did the cell in G1 come from? It came out of division, right? So what happened to the genetic information in division? The chromosomes were torn apart, right? Okay, so got a new cell. It's very small. It's growing. And it got its half of each of the chromosomes. Well, that cell is growing, and it's getting ready to do what? It's getting ready to replicate that DNA, right? Well, the cell wants to check to make sure that when that chromosome was torn apart, that everything is okay before it makes a copy of what's there. I mean, think about it, because in division, if something went wrong, and things go wrong, okay? In division, if something went wrong, if we don't make sure it's okay, we're going to copy it, and then we've got two somethings that's wrong, right? So this is kind of like your English paper, okay? If you're getting ready to make a copy of your English paper, would you want to spell check it first before you made the copy? Because if you don't spell check it, you're going to have the problem or the mistake in both copies, right? So you check it, and then you copy it, okay? So what's going to happen after we copy it? What are we going to do with those two copies? We're going to split them again, yes. Things go wrong in replication a lot, okay? So we replicated before we split the copies and put them into cells alone. Let's make sure everything's okay, because if we've got a problem, then we're getting ready to send that problem to a cell all by itself to be copied and copied and copied. So we definitely want to check and fix the problem if we can before we split our chromosome. And there's all kinds of checks and balances right here during the mitotic division itself. So what happens if the cell can't fix the problem? Okay? Do you remember us talking about apoptosis or apoptosis in chapter 4? Programmed cell death. Cells are programmed to destroy themselves if they can't fix the problem. So if during these checks there's a problem that can't be fixed, these enzymes that the cell has called caspases that are normally held back, they're literally unleashed and the cell is digested. So the cell commits suicide. Better for one cell that is damaged and can't be fixed to be destroyed than for it to continue and make copies of itself, and then you've got lots of cells that are not right, okay? Which is basically what cancer is. So that should make you ask me a really important question. If cells are programmed to destroy themselves, why do we have cancer? Yeah. Well, all of these molecules that are out there keeping the cell healthy and monitoring and checking are all proteins. Think about that for a minute. Where did those proteins come from? The cell had to make them, right? Where's the recipe for that protein? You got it. I love that look on your face. She got it. The recipe, the instructions for making the protein, instructions for making the proteins that are checking your DNA and keeping things, proof checking, and the recipes for making all of those proteins are in the DNA itself. So the instructions are in the very thing it's monitoring. So if you get a mutation, and remember I've said they slip through, if you get a mutation in the recipe to make these proteins that are so important, then you don't have them. We'll talk more about that in just a moment. Okay? You got it. Yes. Okay, so before nuclear division, our chromatin replicates and condenses. We've said that about half a dozen times. I just like to look at that picture of chromatin because it always reminds me of spaghetti. Doesn't chromatin look like spaghetti to you? You thought it was spaghetti? I'm hungry too. I put it up there anytime I can. I love spaghetti. Look at that. Okay, so before nuclear division, chromatin replicates and condenses. And we have this structure that we call a chromosome, right? Okay? Some books actually call this a duplicated chromosome and the chromatin a single chromosome. Don't get hung up on the terminology. Just understand what it is, okay? A chromosome is a replicated piece of chromatin that has condensed, right? So whatever is on this side is also on that side. They're identical to each other, okay? Each side is called a chromatid. C-H-R-O-M-A-T-I-D, chromatid. And because they are identical to each other, they are called sister chromatids. And they are held together by this structure called a centromere, C-E-N-T-R-O-M-E-R-E. Some vocabulary that's really important. Each side is a chromatid because they are identical. They are called sister chromatids, and they are held together by a centromere. Okay? Everybody good? Okay. Remember this picture? Chapter 4. There's your cell, animal cell. Does anybody remember what this structure right there is that looks like a Twizzler? Oh. You can't cover it up, can you? A centriole. Okay. Well, there is this place in the cell called the centrosome, okay? And it is responsible for producing or organizing all the microtubules, okay? It produces the centrioles that we said look like Twizzlers. Again, if anything can be a food item, I can remember it then, okay? Centriole. Okay, centrioles are very important in cell division, extremely important. They are going to produce the protein fibers that are going to be responsible for moving our chromosomes and tearing them apart. Those fibers are called the mitotic spindle. Sometimes we call them spindle fibers. Okay, so the mitotic spindle. Yes. The centrioles produce the mitotic spindle. Okay. Take a look at this for just a moment. This is your cell cycle on an average 24-hour example. Some cells divide sooner than that. Some cells take longer than that to divide. And some cells don't even go through the whole cycle. They arrest at something called G0. But look at this 24-hour cell cycle right there. I love this illustration because it gives you a real sense of how long the cell spends in each one of these phases relative to the others. Okay? So here's your G1. Here's your S, there's your G2, and there's your M. So look how long the cell spends in interphase. Wow. Look how long the cell spends in the mitotic stage. I mean, if this is 24 hours, that M stage can't be more than 2 hours, right? So most of the cell's existence is spent in interphase. And of that interphase, most of it in G1, yes. Look at that. I think this is really important to see because I find that most people come out of high school with a misconception of what mitosis is relative to the life of the cell. I mean, look at it. The cell only spends 2 hours out of 24 in mitosis. The rest of it's in interphase. But we spend so much time in high school teaching that I think most students come out of high school thinking cells live to divide. Okay? Because we emphasize that so much. But think about it for a moment. In G1, it's growing. In S, we're replicating our chromatin. In G2, it's preparing to divide and continuing to grow, right? But what else is it doing? We forget to tell students this. It's growing and replicating its chromatin and growing, but what else is it doing? What's it supposed to do? Exactly, very good. It's got a job to do. So if it's a liver cell in G1, and G2, it's doing its job. It's being a liver cell, whatever liver cells do, okay? If it's a kidney cell, it's a kidney cell, okay? So when we talk about the cell cycle and growing and replicating, blah, 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 keep in mind that the cell has got a job to do, and it's doing its job while all this other stuff is going on, okay? Does that make sense? Okay. Well, let's pause for just a moment, take a little 5-minute break or whatever, and we'll come back in just a moment. Okay, so let's continue talking about mitosis now. We said the mitotic division was divided into mitosis and cytokinesis. Those are two separate events. In most cells, they occur simultaneously. So we'll talk about mitosis first, and then we'll talk about cytokinesis. Mitosis, nuclear division, is divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase or telophase. Okay? 5 parts of mitosis. Now keep in mind, these are more or less benchmarks to a continuum of nuclear division. We have certain events that we say if the cell has done these things that mitosis has progressed to prometaphase. Okay, so these are benchmarks. So let's start with prophase. Before we even get into what's going on in prophase, let's make sure we understand our illustration, because your author, the author of your book, literally uses this same example for the next 3 chapters. So let's make sure we understand the example that we're looking at as we talk about mitosis. This is a cell. The normal diploid number for this organism is 4. So you see 4 chromosomes there. That's the diploid number. Diploid means that there are 2 of each kind, right? There are homologous pairs in diploid cells. So tell me which of those 4 chromosomes, which 2 would be homologous? Which ones look alike? And think about what we said when we said, what does it mean that they look alike? The size, yes.
Like two sizes, you pair them up together.
Yes, so which ones are homologous? The 2 big ones and the 2 small ones. Very good, excellent. Chromosomes don't normally come in red and blue, okay? So don't think of them in terms of red and blue. So those 2 right there, those 2 big ones would be homologous, and the 2 little ones would be homologous. So why do you think the author made one of each of those red and one of each of those homologous pairs blue? One came from the egg and one came from the sperm. That's exactly right. So this would be the genetic information from the egg for that pair of chromosomes. That would be the blue one would be the genetic information from the sperm for that homologous pair. Okay, does that make sense? Okay. Okay, so we're going into prophase, which means that the cell just came out of G2, right? Right, so we've got replicated condensed chromosomes now, right? Okay, so my chromatin has condensed. We just came out of G2. I've got these straw-looking structures right there, the yellow structures in the illustration, that are coming from what looks like 2 earplugs, okay? So these earplug, those little yellow dot-looking things, those are your centrioles. Those are representing your centrioles. And you see that there's 2 of them on the left side of the cell, and you see that these 2 up here at the top of the cell, they're actually moving to the right side of the cell. So the centrioles are going to go to opposite sides of the cell, okay? They're going to produce these structures called the spindle fibers or the mitotic spindles. So these yellow line-looking structures, those are actually illustrating protein filaments, microtubules. That's my mitotic spindle, right there. So those centrioles are beginning to form that mitotic spindle, and the centrioles are starting to migrate to opposite sides of the cell. The nucleolus disappears. Not sure why. A little bit uncertain about that. We're not sure why that disappears, but it disappears, and the nuclear envelope disintegrates. Okay? So basically what we've done is we've eliminated all barriers to our chromosomes. Okay? Because ultimately what's got to happen in the next stage is these spindle fibers have to attach to the chromosomes. They have to reach the chromosomes. They have to get there and attach. So when that happens, we say that the cell has progressed to prometaphase. Okay? Prometaphase, we get these structures that form on the centromere called kinetochores. So we get kinetochores forming. The kinetochore is what's going to attach to the spindle fiber. So we're going to have a spindle fiber from each centriole connecting to the kinetochores of each chromosome. So notice how the short red one has a spindle fiber to this centriole over here to the bottom right, and it also has a spindle fiber to the centriole at the top of the cell. So there's going to be a spindle fiber from each centriole connect to each chromosome. Okay? So the spindle fibers attach to the kinetochores on the sister chromatids. Okay, now we can move them around because those spindle fibers are like bungee cords. They're going to pull and push. Now we can move those chromosomes around. If we tried to split these chromosomes right now, it would be a logistical nightmare. Because think about it. Each half is going to have to be pulled to opposite ends of the cell, and they're going to have to try to go around each other. You wouldn't be able to do it. It would be very difficult. So what happens next is these spindle fibers literally move the chromosomes so that they are very neatly lined up in the middle of the cell, and we call that metaphase. When the chromosomes have lined up in the middle, it's referred to as metaphase. Underline the prefix meta. Meta means middle. So anytime you see metaphase, something's in the middle. So look how easy it's going to be now. Now that we have everything organized in the middle, these spindle fibers just have to pull toward their centriole. So these would pull downward, coming toward this end of the cell, and those up there would pull upward, going to the top of the cell. And that would literally pull those chromosomes apart. When that starts to happen, when the sister chromatids start to separate, that is anaphase. The sister chromatids split, and each half is pulled to opposite pole. That's anaphase. Okay, so they get to the opposite pole, and it's telophase or telophase. Okay, the way I want you to think about telophase is we've got to form two new nuclei. That's what mitosis is, right? Producing two nuclei. So everything that's going on in telophase is the opposite of prophase. Look at what happened in prophase. We're going to undo all of that because we got to make two new nuclei, okay? So in telophase, our spindle's going to disappear. Remember, it showed up in prophase, right? So it disappears in telophase. Our new nuclear envelopes are going to form around our two nuclei. Our chromosomes are going to become diffuse chromatin again, single-stranded, and our nucleoluses come back. So that is telophase. And then we have two new cells. That's not bad. Not too bad, yeah. So let's talk about cytokinesis for a minute, because we said cytokinesis is a separate event for most cells that occurs simultaneously, but it doesn't have to. Cytokinesis is division of the cytoplasm. It begins in anaphase. It will continue through telophase. and it's completed at the interphase. And remember, mitosis without cytokinesis will result in a multinucleated cell. So, how does it start? Well, it's going to start with this structure right here. This is called a cleavage furrow. Has anybody ever gardened and furrowed a garden? Do y'all know what furrowing a garden is? It's when you take a plow and you go through and you make a trough, and that's where you put the seeds. That's a furrow. You furrow a garden. So cytokinesis starts with the cleavage furrow right here. Should be easy for y'all to remember. That's the cleavage furrow, okay? And these proteins, this contractile ring, literally squeezes the two cells in half, okay? Have you ever taken Play-Doh and held it in your hand and squeezed it and it come out both sides? That's what's happening to the cell. So with time, that contractile ring will make it look like that, and your cells will be pulled apart. Okay, plant cell division. As far as the steps for mitosis, very similar. The one thing that you probably need to know that's different in plant cell division is it occurs in tissues called meristematic tissues in plants. We sometimes call them meristems. Meristems are the part of a plant from which it grows. Okay, and all growth is mitosis. I mean, growth is adding more cells, right? How do you add more cells? Mitosis. So those areas of the plant that grow all the time are the meristems. So where do you think you would find those? From where does a plant grow? From the roots and the shoot, the tender little leaves at the top. So the meristems are found at the roots and the stems, and mitosis is occurring here all the time. So you have very similar, what you're seeing here is the stained plant cells in division. The upper left is prophase. You see replicated condensed chromosomes. Bottom left is metaphase. They're lined up. Top right is anaphase, they're pulling apart, and then bottom right is telophase. You're getting two new cells there. One thing that's very different in plant cell division is cytokinesis, division of the cytoplasm. Plant cells can't furrow. You can't have that indentation. You can't furrow a plant cell. Why? Very good, because of the cell wall. Excellent, yes. Plant cells have a rigid cell wall, and it doesn't permit furrowing. So what you end up happening are these vesicles right here. This is going to form what's called the cell plate, and those vesicles contain the components of the new cell wall right there. So you have a cell plate that forms. You can't furrow if you're a plant cell. Okay, any questions? Let's watch this short little video. It's very short, it's very simple, but it's good.
Mitosis begins as the long threads of DNA in the nucleus start to coil. Having already replicated, these threads emerge as the double strands we know as chromosomes. Meanwhile, protein fibers grow from the migrating centrioles, forming a latticework of spindles. For reasons scientists don't fully understand, the nuclear membrane suddenly disintegrates. With amazing accuracy, a spindle from each centriole attaches itself to each of the chromosomes. Assisted by the protein fibers, the chromosomes move center stage. In a microscopic tug of war, the spindles pull each chromatid toward opposite poles. Other spindles push against each other in a ratcheting action. that stretches the cell. The chromosomes then unwind, and the nuclear membranes reform. With mitosis complete, the nuclei and cytoplasm separate, creating two new cells in this ongoing cycle that sustains all life.
Okay? How many people are going into micro? What in the world? Maybe not. Crazy. How many people are going into micro? Anybody going to take micro? How many people have taken micro? Okay. Okay, so let's talk about prokaryotic cell division for just a moment. We can't call prokaryotic cell division mitosis. We have to call it binary fission. Why do you think that is so? Why do we call it binary fission? Why can't we call cell division in bacteria, prokaryotic cells, mitosis? What is mitosis? What definition do we give? Mitosis is division of the nucleus, okay? What's the definition of a prokaryotic cell? It has no nucleus, so we can't call it mitosis. They don't have a nucleus. It's called binary fission. Prokaryotic cells divide by binary fission. Binary, bi means two, okay? Fission means to split. So we are literally splitting in two, okay? Can't call it mitosis because these cells have no nucleus. They also don't have a whole lot of those other organelles that we talk about. And they have one single circular chromosome. What's the area called where that chromosome exists? The nucleoid. Yes, very good. So here it is. Let's take a look at prokaryotic cell division for just a moment. On the upper part of the illustration, you see your prokaryotic cell there. There's your single circular chromosome. Keep in mind that prokaryotic cells have an envelope that consists of an inner layer called the plasma membrane. They would then have a cell wall and a glycocalyx. Remember, we called it the envelope. Okay? So the first thing that happens in prokaryotic cell division or binary fission is that chromosome is going to attach to the top, to the inside of that plasma membrane. So now my chromosome is attached. It replicates itself. So in this cell right here, we have two copies of that single chromosome. The cell starts to elongate, and as the cell pulls, it's going to pull that plasma membrane, which is going to separate those two copies. So you end up with the two copies being separated. You have the cleavage, cytokinesis, and then you have your two cells, each with a copy of that single chromosome. Very simple. So what happens when cell division goes wrong? You end up with something called cancer. Cancer is a growth disorder, or is defined as a growth disorder that results from the mutation of genes regulating the cell cycle. So guys, cancer is not a foreign invader. Cancer is your own body. Cancer is your own cells where this cell cycle has gone haywire. It's like, normally you would go put a copy in the Xerox machine and press the 2. You want 2 copies and hit the button and you get 2 copies out, right? That's what your cell cycle is supposed to be. Well, in cancer, you put 2 in and you press start and the machine gets stuck and you get copy after copy after copy after copy, and that is cancer. It's the cell cycle out of control. That's why it's so hard to combat, is because it's your own cells that have gone haywire. It's that cell cycle out of control. So what does it mean when we say results from the mutation of genes that regulate the cell cycle? Well, remember, genes are recipes for proteins. So what we're talking about are those proteins that are involved in cell cycle regulation. So you've got a mutation in those recipes, those genes, and those proteins aren't regulating the cell cycle. The formation or the beginning of cancer is called carcinogenesis. The development of cancer is carcinogenesis, and it tends to be gradual. Let's talk about that for a moment. The development of cancer is carcinogenesis, and it tends to be gradual. You've probably heard of a carcinogen. Those are substances that cause cancer, okay? Tends to be gradual. Let's talk about that for just a moment. I'm going to stand here, and I'm going to say that from here until the edge of the desk is a continuum, okay? And the edge of the desk is cancer, okay? So let's say that I'm talking about a certain gene involved in cell cycle regulation, and at this point, this far away from the desk, that gene is perfect. It's correct. The recipe is right, okay? The A's and T's and C's and G's that make up our double helix are all right, and they're in the right order. And it's coding for the exact perfect sequence of amino acids that cause that protein to form, that three-dimensional structure, okay? So let's say I start right here. I have the perfect recipe. Well, guys, every time your cells divide, changes slip through, mistakes slip through, okay? Even with all the proof checking, everything that's there to make sure that your cells are healthy, there are always some changes in the A's and T's and C's and G's that slip through for one reason or another, okay? So every time your cells divide, you get changes to your DNA. Well, most of the time it doesn't matter because, guys, my chromosomes, my 46 chromosomes, are made up of over 3 billion base pairs. Okay, a base pair is the A and T of the double helix. You guys remember that when we talked about it in Chapter 3? A base pair would be a C and a G. Remember, that's complementary base pairing. Well, there's 3 billion of those. And only 1% of 3 billion, think about that, 1% is involved in coding. So your genes, we know where some 25,000 genes are. Only 1% of those 3 billion base pairs make up those 25,000 genes. Okay? So you got 99% of 3 billion that if a change occurs, it's no big deal that we know of. Okay? So as your cells divide, changes occur. Okay? So I've changed, right? I'm not over there anymore. I'm over here. So let's pretend this is a real recipe. Let's use an analogy of a real recipe, what just happened, okay? Let's say that over there, in that perfect slot, I'm making a pound cake. You culinary people are going to love this. I'm making a pound cake from my great-great-grandmother's recipe, and it calls for Swan's Down cake flour. Do y'all know what that is? The whole right side of the room is going, yes, yes, yes. Swan's Down cake flour is this really expensive cake flour. It makes the absolute best cakes ever, okay? So this recipe is calling for that, and it's fine. I mean, that cake is fine. Okay, over here, I've got some changes to my recipe, and now it's calling for great value self-rising, okay? Nothing against great value, but it's not Swan's Down. Okay, so is it still a cake? Yeah, it's still a cake, right? Is it as good as that cake over there? No, okay, but it's still a cake. So at this point in time with my DNA, it's still functional. I mean, I'm still coding for a functional protein, okay? And then all of a sudden, you know, I'm being exposed to toxins and carcinogens as I live. Whatever, and my DNA is changing more. And please keep in mind that as these changes occur to my DNA, it's changing the amino acids in my protein, right? And so those relationships that we talked about in chapter 3 that cause that protein to take that three-dimensional shape that we talked about, those relationships have changed. Okay? But somehow it's still functioning until one day all it takes is one more change and the protein's not functioning anymore. So you just fell off the cliff. That's cancer. Okay? But keep in mind, guys, it was not that one change. It was the accumulation of all of those changes that caused this to happen. And the scary part, that's a nice little story there. The scary part is we don't start right here. You don't know what you inherited. You could have inherited this, or this, or this. Your sequence, you could have inherited this right here. If you inherited a sequence in a gene that is this close to the cliff, that's called a proto-oncogene. Proto means just before becoming an oncogene. An oncogene is a gene that causes a cancer. Okay? A good example of a proto-oncogene that you may be familiar with is BRCA1. Okay, anybody ever heard of that? Breast cancer. We call it the breast cancer gene. It's actually not the gene that's causing the cancer. It's the sequence in the gene. Okay? So if you inherited that sequence in that particular gene, then you are really close to the edge. Now, does that mean that you're going to have cancer? No, not at all. That means that your sequence can accommodate less changes until you fall off the cliff. Okay? Normal individuals, if they find out, if you have a history of breast cancer in your family, I strongly recommend that you be sequenced to find out if you have this sequence, because then you monitor all the time. Okay? And most people, monitor and do fine. Some people take drastic measures, like Angelina Jolie. Y'all know who that is? All the guys are going, yeah, we know who Angelina Jolie is. Beautiful woman. She found out that she had BRCA1. What did she do? She had a radical double mastectomy. Yes. I think did she even have a hysterectomy? I can't remember if she did or not. But most people would just monitor. Okay? Any questions? Before we only have like 2 or 3 more slides. I meant to ask this earlier. I want to see, just out of my own curiosity, if you have been touched in some form in your life. Doesn't have to be a family member, just somebody that you care about, by cancer. Raise your hand. Stand up in honor of these people, or in memory of these people. For some of you, stand up. I want to see. Unbelievable. Yeah. Yeah. Unbelievable. Thank you. So let's get all that in your notes. Origins of cancer, mutations in DNA repair mechanisms. That's what we've been talking about. Those proteins that are there to ensure the safety and the correctness of your DNA. If you have mutations to those proteins, the genes that code for those proteins, you don't have those repair mechanisms. Okay? Mutations to proto-oncogenes, where they become oncogenes. And guys, we even have molecules in our body that suppress tumors, but they're proteins. So if you have mutations to those genes that code for those proteins, you don't have them. And to make matters worse, guys, every time a cell divides, the end of the chromosome, that's called the telomere, the very end of the chromosome is called a telomere, and it's always a non-coding region, meaning it doesn't have a gene in it, but it's very important functionally, okay? It has to be there. The end of the chromosome, during every cell division, falls off. And there is an enzyme called, the end is called a telomere. Okay, tele means end. The end is called a telomere, and there's an enzyme called telomerase that's supposed to build it back and make it right again. Well, if telomerase is not functioning correctly, it allows that cell to continue to divide. Okay? So telomerase, another protein enzyme, very important. Guys, there's actually, well, every time your cells divide, the telomere falls off. Telomerase is supposed to build it back, but it never builds it back just right. Okay, so it gets shorter and shorter and shorter as we age. We think that's probably one of the reasons we age is because our telomeres are shrinking. There's a company out there called Years, and I swear I'm going to do this. But you can spit in a tube because your saliva contains cheek cells, so they can take your saliva and sequence your DNA and analyze your telomeres. And they can send you a report back to give you an estimate of how you're aging. So you can see, are your telomeres shrinking at the same time that you're at the same as you're aging? Are you aging physiologically the same that you are chronologically? I mean, I may be 60 years old, but if my telomeres are 40, hey, that's good. But then again, I may be 60 and my telomeres may be 80. Okay, that wouldn't be good. Okay, but I swear I'm going to do that. But they tell me I'm 80, I'll probably go into depression. Okay, so cancer is, I want to do it though. You can spit in a tube and find out all kinds of good things. I will talk about that more in other chapters. Cancer is over 100 different disorders. Okay. Keep in mind that liver cells are different from kidney cells, that are different from colon cells, okay? So the cancer in a liver cell is going to be different than a cancer in a colon cell, okay? Usually you can tell what the cell was before it became cancerous. But over 100 different disorders, but they all have some things in common. Even though they're different, they all have some things in common. So let's talk about some of the commonalities of cancers. All cancer cells lack differentiation, okay? Differentiation is something that occurs as a cell develops. Cells start out as like generic cells, they're stem cells, okay? They haven't decided what they are yet. And they develop and differentiate into a type of cell that has a job to do. And once cells differentiate, they don't go backwards, okay? Once you're a liver cell, you're stuck. You're a liver cell, okay? Normally they don't go backwards. Cancer cells have gone backwards. They are differentiated cells that have lost that. They don't know they have a job to do anymore, so they don't do their job anymore. They also have abnormal nuclei. It's misshapen, it's larger. It's not right, okay? They also have the ability to form tumors, okay? They have lost what's called contact inhibition, okay? Let's talk about that for just a moment. They've lost contact inhibition. We said earlier that normal somatic cells divide for two reasons. What are they? Growth and repair, that's right, okay? So you grow and you grow and you grow, and once you're grown, your cells will divide when other cells die and need to be replaced, okay? If you take normal cells and you put them in a dish and give them lots of nutrients, they're going to divide and divide and divide and divide until they fill up the dish, okay? And then they'll stop dividing. They come in really close contact with each other. It's called contact inhibition. And they will divide after a cell dies and needs to be replaced. Okay? That would be growth and repair. Cancer cells don't do that. You put cancer cells in a dish with lots of nutrients, and they divide and divide and divide. And when they come in close contact with each other, they don't know they're supposed to stop. They divide and they grow on top of each other. They even grow out of the dish. Okay? So they've lost that contact inhibition. Cancer cells also have the ability to undergo metastasis. You've probably heard of people's or individuals' cancers metastasizing to a different location. This is the ability to develop a tumor distant from the primary tumor. New tumors distant from the primary tumor. Keep in mind, a colon cell that develops cancer is going to be different from a liver cell that develops cancer, okay? So if you've got colon cancer and a cell breaks off and travels to the liver, which seems to be a common route, and sets up a tumor in the liver, that patient would not have colon cancer and liver cancer because that was not a liver cell, Okay? Medically, that patient would have colon cancer, and colon cancer that metastasized to the liver. Okay? And you ladies could probably teach this better than I could. Okay, undergo metastasis. They also have the ability to undergo angiogenesis. What is angio? Angio. What's angio? Blood, yes. What's genesis? Huh? Genesis? The beginning, yeah. So, yeah, I mean, just guess, yeah. Angiogenesis, they have the ability to make your body form new blood vessels. Okay? Think about it for a moment. Well, first of all, let's back up for a minute, guys. Your vascular system is formed during development. Okay? Yeah, tell me if I'm saying something wrong, guys. Your vascular system is formed during development. Once you are developed, normal cells don't build new blood vessels unless you're injured, okay? And then your body would build new blood vessels. So normally your body, once you're developed, you don't form new blood vessels. But these cancer cells have that characteristic of the cells that are developing. They have the ability to tell your body, make me a blood vessel. Think about it for a moment. This is a tumor. It has to be fed. It needs oxygen. It needs nutrients. It has waste products it has to get rid of. If we could combat that characteristic of cancer cells, you could starve them to death. This is HeLa cells. Y'all know, ever heard of HeLa cells? Anybody know what HeLa cells are? You ladies need to know what HeLa cells are. Has anybody ever read the book Henrietta Lacks, The Immortal Life of Henrietta Lacks? Henrietta Lacks was a woman who lived in the early 1900s in New England, and she had cancer. Was it cervical? Yeah, I think it was cervical cancer. Anyway, she was very, very poor. This was in the early 1900s, guys, before any kind of privacy law. And it was common practice that when people went into the hospital with tumors, that it was just understood that the medical people were going to take a part of that tumor and they were going to experiment with it. They were going to try to grow it and do tests on it. They didn't even ask you, okay? They just did it, okay? So Henrietta Lacks goes in and that she has parts of her tumor removed, and she has her tumor removed, and then they take part of it to the lab. And they always named the tissues after the patient. The tissues were always named, the cancer tissues were always named with the first two letters of the patient's first name and the first two letters of the patient's last name. And her name was Henrietta Lacks. So it was H-E-L-A. So her cells were called HeLa cells. And you may say, well, why did they want these cancer cells? Well, they were studying them, obviously, but one of the things they wanted to do was scientists were trying to find a cell line that never died out, okay? When you're doing experiments, we talked about experiments in Chapter 1, when you're doing experiments, it's very important to keep everything the same and only have one experimental variable, okay? So if I'm testing a drug, the effects of a drug, then I need to use the same cells, okay? Because if I don't use the same cells to test the drug, I won't know if the effects are from the drug or the different cell, right? So that's really hard when you're working with tissues because cell lines die out. A cell line is when you have a cell and it's little and it grows and it divides, and then those two cells grow and grow and grow and they divide, and those two cells grow and grow and grow and they divide. All those cells that came from that initial cell are called a cell line, okay? And cell lines don't last forever. They eventually, all of them die out, okay? Yeah, they don't last forever. But what happened with Henrietta Lacks is something that still to this day nobody understands. Her cell line, her cancer cells never died, ever. We have no idea if it's her cells or her cancer, whatever it is, her cells never die. Henrietta Lacks died shortly thereafter. Her cells have become a multi-trillion-dollar international enterprise. Every drug that has ever been produced since the 1900s has been tested on her cells. When a scientist is doing experiments and needs to work with tissues, they order HeLa cells, and they're very expensive, okay? So her cells have produced massive, I can't tell you how many tons of cells her cells have produced. I mean, it's a massive industry because they never die out. And the sad part about it all, and the only reason I tell you this, is because I feel like is an injustice. Her family did not get a dime out of any of that. So they were here about 4 years ago. Anybody hear them? Her family was actually here on a book tour. The guy who wrote the book and the family was here. Anyway, now you know who Henrietta Lacks is. So if you, they did a movie about a year ago. I did not like the movie at all. The movie was more about, not even so much about Henrietta Lacks. I didn't feel like it did the science justice. Anyway, I think that's it. Hang on 2 seconds.